Okay, welcome everyone. And so this is, believe it or not, the 24th CCHF Virtual Symposium. And today we've got a fantastic group of speakers. It's going to be a really exciting event. We're going to start off with Ming-Yu Ng from Stony Brook University, then Laurel Schaefer from the University of British Columbia, and then Ming-Yu Ng from the University of Toledo. And I'm really excited to hear these three presentations. It should be a really great event. Okay, if I can have the next slide, Rio. So one of the things we'd like to encourage is a little bit of group engagement. One of the problems with Zoom, of course, you often listen to people, and how do you connect? So Rio, our managing director of the CCHF, has brought in this mentee for us to be involved in being able to interact with each other. So Rio, why don't you take the lead and explain to us how this works?
Oh, well, it's very easy. Just basically go to menti.com using either your browser on your phone or in your computer and enter the code 55708211, or you can also scan the QR code using your phone camera, and you'll see the presentation as you go. And there's a surprise for you. Actually, there's a new style of the question for practice. Whenever you're ready.
So Rio, we're having a little bit of a difficulty here with the internet at Emory. So a few might not be able to get to Menti. For some reason, my computer is working, but many of the other internet facilities are not. So we'll see how many people can actually join Menti at this stage. But encourage people to sign up if they can. For example, my iPhone has gone out of action at the moment. I don't know what's going on here.
Yeah, well, are we ready for the next slide, Hugh?
Yes, you go ahead.
Okay. So this is the question, practice question to say where you're from. Yep, exactly. Basically, you can use your phone and put your PIN and submit, and that would add into the map that we have. And we will be using this feature later on for appreciation for our speakers as well.
Oh, we're getting some interesting distribution. And as I said, the internet's not working properly here. So there's locally, there may be some challenges.
Looks like people from Atlanta is logging in though.
Yeah, but they may not be in the building. I think it's a building issue.
All right, whenever you're ready, Hugh.
Okay, so that's great. That's a good start. Okay, so let's move on. Next slide. Okay, so we started these virtual symposia about 5 years ago, and we thought we were very clever back then because we built this center without realizing, the US is a big country. I come from a very small country, Wales, so it's very easy to cover the whole country and people can go on bicycles from one end to the other. But when we built this center about 10 years ago now, we had 22 professors from 15 universities, and we had to learn how to do large-scale communication. And of course, we were very proud. We actually got involved in an early version of Zoom and even an earlier version of large-scale communication. And we were very proud of ourselves. Of course, now everyone knows how to do it, so it doesn't mean anything anymore. But because we were able to communicate effectively, we decided to go with these large-scale virtual symposia. And that's been very exciting, as you can see from the next slide. Yeah, we tend to meet every year as a center, but actually we have the virtual meetings once or twice a week. People thought I was a total megalomaniac insisting on such common meetings, but they've been very effective for us. Next slide. And as a consequence, we started these virtual symposia. So it's been great. We've had about 3 or 4 of these a year. And here are some of the early ones. The next slide, please, Rio. And we've continued this. We started off with 4, but I think that got a bit too much for people. It took a little bit too long. And then we went to 3 speakers per session, and that was a more comfortable zone for everyone. And we've also done virtual symposia. Our last 2 were actually on alumni that had come out of the CCHF, and those were tremendously exciting as well. And so now with the 24th, we're going back into some of the real exciting groups that are involved in siege functionalization. Rio also wants me to mention, next slide, please, that we've actually supported the Division of Organic Chemistry to run much broader symposia in the field of organic chemistry beyond CH functionalization. And of course, CH functionalization is still very important. As you can see, Hans Renata is one of the speakers at the next one, as well as Mark Levitt. So that's coming up pretty soon, February 16th. So I'd encourage you to think about joining that. That's on the Wednesday and starts at 12 o'clock Eastern Standard Time, not the 4 o'clock Eastern Standard Time of our typical meetings. Okay, next slide, please. Oh, yes. So the CCHF is starting a new initiative called Beyond CCHF. And so as a consequence, we are hiring an associate manager to be involved with that. So if you're interested in this, again, click on the code and or contact us because we're very excited to get someone who's interested in this large-scale, synergistic, collaborative type of environment to help manage this new initiative. Okay, next slide, please. Okay, so to the main piece of action, which is our 3 wonderful speakers. So we're going to begin with Ming-Yu Ng. So let me just quickly introduce Ming-Yu. So Ming-Yu actually began doing undergrad research in catalysis from the University of Hong Kong in 2003. He then went to do his PhD with Mike Crochet at University of Texas at Austin, and then did two postdocs, one with Barry Trost and then with Tobias Ritter. And so a lot of focus throughout his developmental career in catalysis. And so then he began as an assistant professor at Stony Brook in 2013 and was promoted to associate professor in 2019. And as far as I'm concerned, his program is really extremely creative in the way it approaches aspects of CH functionalization. Obviously, I've seen a lot of CH functionalization by now, but I was tremendously impressed with the creativity that's gone into his research program. So I'm very excited to hear about your latest work, Ming-Yu.
Okay, great. Thank you so much, Hill, for your introduction. And let me share the screen. Let's see. Right. Okay, cool. So can you see it clearly?
Yes, we can.
Okay, great. Yeah, thank you so much, Hill, for your introduction and also for giving me this opportunity to share some of the research programs that we have in our lab. And so before I jump into today's talk, I do want to, let me just change it to the, all right, I do want to give a very brief overview of what we are doing in our lab. And so in my lab, what we are interested in is trying to develop some new molecular editing technologies that will allow us to effectively and selectively functionalize organic compounds, because many of the medicines, agrochemicals, imaging agents, and polymers are organic molecules. And so if we can selectively and efficiently functionalize them, then we'll be able to create a wide range of new functional molecules and to advance this field of research. And in my lab, what we are mainly focusing on are 3 major programs. One is doing the molecular editing using the excited state catalysis. And so in this particular program, what we do is that we'll bring the catalyst from the ground state to its excited state using the visible light. And then by accessing to this excited state reaction landscape, what we are hoping to do is that try to develop and discover some new chemical reactivity and transformations. And then we're also doing some molecular editing of carbohydrate. And in this particular programs, what we do is that we're developing new reaction transformations, the transformations that will allow us to selectively functionalize carbohydrate. We also work on the molecular valorizations and using the fluorine chemistry. And so in this area, we developing new reagents and also chemical transformations that will allow us to do the CH trifluoro and difluoro methacylation reactions. And that can also be applied to some complex and late stage functionalization of pharmaceuticals and also agrochemicals. So in today's talk, what I will mainly focusing on will be on this molecular editings of carbohydrate. So why carbohydrate? So carbohydrate is the most abundant organic compound in nature. And so it's not only just provide us the energy source, but it also covered on the cell surface. And so these are the carbohydrate units that join together, oligosaccharide, and we call it glycan. And so these are conjugate onto the protein. And so we call this a glycoprotein and also conjugate to this lipid, we call it glycolipid. And so these glycans, they are encoded with different biological functions. For example, they can use for the cell-cell interactions, communications, and also disease progressions. I'm sure by now, everyone know about COVID. And so on this COVID virus, they also, on the surface of this virus, they also have a lot of like spike glycoproteins. And they use these glycoproteins to bind onto the ACE2 receptors of the host cell and then help to internalize of this virus into the host cell, and then they can multiply. And also over 20% of the natural product, they are glycosylated. And this carbohydrate units play a very important role in their biological activities. So the questions that we are interested in answering is that what will happen if we can selectively or to modify or replace some of this carbohydrate units on the cell surface or on this natural product? Can we alter their biological functions? So in order to answer that questions, it's quite difficult because we know that modifying the carbohydrate is very labor intensive and time consuming. To give you one example, so this is 402-deoxyglucose, and this is one of the very first radioactive imaging agents that's approved for the positron emission tomography imaging. And it has been widely used for the diagnosing cancers and also neurological diseases such as Alzheimer's disease. However, to make this type of molecule is actually quite lengthy. So to make this molecule, what you do is that you will start with this D-mannose, and then you will go through a potassium and then brominations to form this 1-bromo sugars. And then you do the cyclization to form this ortho ester, followed by selective cleavage and to free up this 2-hydroxy group. And then you will activate it to convert it into the tryptophan. And then you do the fluorination and depotassium and to give you this product. So basically, in order to replace one of the hydroxy group on the carbohydrate with a fluorine atom, it will require you 7 steps and less than 14% overall yield. So as an organic chemist, I would like to ask the questions, can we develop a more efficient and general strategy that will allow us to site selectively functionalize the carbohydrate? And if we can do that, then it will likely going to accelerate the development and discovery of the carbohydrate-based therapeutics, vaccines, and also the imaging agents. And it also allows us to create some new tools to answer some fundamental questions in glycobiology. So what we start with is that we want to focus on this C2 positions of the sugar. And the reason why we are interested in that class of the glycomimetics, because many of them has been applied to the medicines, imaging agents, developments, and also cell service engineering, and also in catalysis. And so in order to seek the ideas, so we come across a very interesting paper reported back in, reported by guys back in 1987. So what he did is that he subject this 1-bromo sugars to the tin hydride and AIBN conditions. And what he observed is that there's a reduction of the C1 position. So this is not very, this is expected reactivity, so it's nothing surprising. But what is unexpected is that when he do the slow addition of the tin hydride using the syringe pump, what he observed is the C2 reduction of the carbohydrate. And so the mechanism that they propose is that under this tin hydride conditions, you generate this tin radical, which abstract the bromine atoms from this 1-bromo sugars to form this C1 radical. And this C1 radical will then undergo a process called a 1,2 spin center shift. Basically, it will undergo a conformational change followed by this acetoxy group migrations. And now you have access to this C2 radical. And this C2 radical will then take a hydrogen atoms from this tin hydride and to give you the desired product. So the questions that we ask is, can we capitalize this particular reactivity and to develop a wide range of the reactions that will allow us to introduce different functional groups at the C2 positions of this carbohydrate? And in order to do that, what we will need is that we'll need a catalyst that allow us to generate this radical, and then a catalyst that can capture this C2 radical for the cross-coupling reactions. And when it comes to the cross-coupling reactions, I'm sure palladium is doing a really good job. And so in this ground state palladium catalysis, where the palladium would do an oxidative insertion into this alkyl halide bond, go through this T electron process and generate this closed shell species. And this closed shell species, which means that there's no radical or unpaired electrons of this in this compound. And so this can undergo a wide range of cross-coupling reactions, and then you can introduce different functional group into the R group. So this reaction is very important, and it received a Nobel Prize in 2010. But this closed shell reactivity is not very useful to achieve our desired reactions. I mean, you can generate the radical using this by heating the reaction up to a well over like 100 degrees Celsius. Then you can access to the radical reactivity. But thankfully, in 2016, Gawagens, they observed this excited state palladium catalytic activity. Basically, what they do is they're shining light onto the reaction mixture, and they observe the palladium can undergo a 1-electron process going through a hydrogen atom extractions to form this radical and form this open shell get access to this open shell reactivity. And this is in equilibrium with this palladium-2 compounds. And so this is basically, you can convert this closed shell reactivity back to the open shell reactivity just by shining light onto it. And so Gawagen take this reactivity, and then they develop the palladium catalyzed desaturation reactions, and also the palladium catalyzed Hack reactions to incorporate the functional group onto the alkene. So we ask, can we take advantage of this excited state palladium catalysis and then to realize our desired reactions? So here's the idea. So you take this palladium, you photo-excite it to the excited state, which undergo the hydrogen atom extractions to form this C1 radical, and that can undergo a 1,2 spin center shift to go to the C2 radical. And then this will be in equilibrium with the palladium-2 compound. And we are hoping that we'll be able to capture this and then use it for the cross-coupling reaction to introduce different functional group at the C2 position. So my postdoc and also my grad students worked really hard over the COVID time, and they were able to achieve the goal. So now what we can do is that we can take this readily available 1-halo sugars, and then with some coupling partners under this excited state palladium catalyzed conditions. And using this 1,2 spin center shift reactivity, we were able to incorporate these hydrogens, deuteriums, and iodines at the C2 position. And the reactions work for the monosaccharide. It also work for the disaccharide and some of the natural product or drug glycoconjugates. And so for these reactions, one thing that we are very interested in, so we are interested in really understand better about this 1,2 spin center shift process. So this is really the key steps in our reactions. And so this step actually has been studied in the past by guys. And so there are 4 possible pathways. And so one is that it can go through this complete dissociation forming this carboxylic radicals. And then the second pathway is that you are forming this 5-membrane and then putting a radical at this carbon. And then the third pathway is that you have the dissociation, but it's more like going through the ionic type dissociation, or it's going through a concerted mechanisms and to have this radical migration. And so we were very interested in just to understand which of this reaction is actually going on in our system. And so we did some preliminary like mechanistic studies and then trying to rule out some of these pathways. So for the first pathway, we know that if this decarboxylation process is really, really fast. So if we do generate this, going through this pathway 1, it will likely going to form this methyl radicals instead of like undergoing through this migration. Therefore, what we believe, because of the rate of the decarboxylation is very fast, is much faster than the rate of migration. Therefore, the pathway 1 is very unlikely. And for the pathway 2, we also did some experiments like to understand whether this is possible or not by incorporating the radical clock into that. And so if we do generate a radical at this carbon position, it should open up the ring and to give you this ring opened product. But we didn't observe that. Instead, we just observe our regular desired product. And so that also implied that the pathway 2 is also very unlikely. For the pathway 3 and 4, what we did to distinguish them is that we did some crossover experiments. And our hypothesis is that if it does going through the full dissociations, that could see some crossover. So we run the reaction using these 2 different substrates, but unfortunately, we didn't observe any crossover product. So what this result indicate is that it could be the pathway 3 going through some in-cage recombinations, or also support the pathway 4 process, the concerted mechanism. And so we take advantage of another mechanistic studies. And so if it does going through this dissociation, forming this anion, then by changing the electronics on this R1, we should expect to see some change in their rate. So we perform the reactions and then using the electron withdrawing group and the electron donating group. But what we see is that their rates are quite similar to each other. So based on this data at the moment, what we believe is that it's likely going through this P4 pathway for this key step. So with the understanding of this 1,2 spin center shift, we want to broaden the scope of these reactions. And so the questions that we ask is that can we use other catalysts or other metals to expand the scope of these reactions? And we look at the periodic table, and I think nickel is a very good choice. It's not just because nickel is like 5,000 times cheaper than palladium, but it also have a lot of properties and reactivity that is very useful for our desired reactions. One of which is that it can undergo at both 1 electrons and 2 electrons reactions, and to also access to this alkyl radical. You also have a really broad cross-coupling profiles that will allow us to incorporate different functional group onto the carbon. And back in 2004, and Grateful reported the nickel catalyzed radical Suzuki-Miura cross-coupling, where he take this alkyl bromide and then using this nickel catalyst and also phenylboronic acid. And then what he observed or proposed is that it's going through this alkyl radical and then followed by recombination, reductive eliminations to give you this desired product. So the questions that we ask is that, can we develop a C2R relations using this nickel catalyzed migratory Suzuki-Miura cross-coupling reactions? So basically you take the 1 bromo sugars with the nickels and aryl boronic acid, and then you can go through, form this C1 radical, go through this 1,2 spin center shift, recombine, and to incorporate this aryl group at the C2 position. So the postdoc and grad students also work day and night in the lab, and we were able to get this to work. And so now what we can do is that you can take this 1 bromo sugars, and then with this aryl boronic acid and then nickel catalyst, and then you can incorporate this aryl group onto the C2 positions. And you can also use some heteroarenes as well for this particular process. And different sugars, monosaccharide, can also be used under this condition. And the migrating group, we also look into that, and they are tolerated as well. And here we also use some natural product glycoconjugate or some disaccharide or some the drug glycoconjugates, and they all work pretty nicely under this condition. So we did some mechanistic studies, and we also trying to do some computational studies as well. And so currently there's two major mechanistic pathway that we have in mind. One of which is that you can, so under the reaction conditions, we generate this nickel-1 bromide, and then this will undergo a ligand exchange and transmetallations basically, and then you form this nickel-1 aryl complex. And this nickel-1 aryl complex will then undergo a halogen atom extractions to form this C1 radical. And then this C1 radical going through the 1,2 spin center shift and followed by recombinations of the nickel complex. And then you will be able to do the reductive eliminations to give you the desired product and also regenerate your catalyst. And so in this case, when you have the, you have the C1, this can also recombine with the nickel center, and then to give you this C1 bond nickel species. And this can also undergo a reductive eliminations to give you the desired product. And we do observe some of this byproduct in our reaction mixture as well. And so this is one of the pathway that we have in mind. And there's also another pathway which is going through first the halogen atom extractions and to form this serine radical and form this nickel-2 bromide. And this nickel-2 bromide can then undergo the transmetallations to give you the nickel aryl species. And then this C1 radical undergo the radical 1,2 spin center shift, and then recombine with the nickel reductive eliminate to give you the desired product and regenerate the nickel catalyst. And so for this one, to summarize this is that, I mean, now we can do this C2 arylation using this nickel catalyzed migratory cross-coupling reactions. And this reaction is the first examples where the nickel catalyzed Suzuki cross-coupling that's going through this 1,2 spin center shift reactions. And it also allows us to access a wide range of this C2 arylated sugars derivatives, which is very, very difficult to synthesize in the past. And our hope is that, I mean, this new compound will potentially, we can explore their biological activities. And so what I have told you today is that we take advantage of this 1,2 spin center shift reactivity, and we were able to use the palladium catalyzed reactions, and then we're able to incorporate these hydrogens and also deuteriums at the C2 positions. And also we can introduce the iodine at the C2 position as well. And using the nickel catalyzed conditions, we also able to incorporate the aryl group at the C2 position. So we also able to take the alkenes to do the C2 alkynylation. And I didn't get a chance to talk about it here. And that one, you can take just a simple alkene and styrene, and then you can do a CH glycosylate, CH functionalization to add these sugars at this C2 positions. And so our hope is really like continue to develop more molecular editing technologies that will allow us to modify carbohydrate so that we can take these simple and readily available sugars and convert them into medicines, imaging agents, and biomaterials. And we also want to generate new biochemical probe that will allow us to really addressing and answering some fundamental questions in the glycobiology. And with that, I would like to thank my wonderful team of postdoc and grad students and undergrad students they work really hard in the lab and trying to push the boundaries. And it is a science and really pushing the science forward. And so the sugar chemistry is mainly done and started by my postdoc, Gaoyuan Zhao. And then there's also 3 grad students also working with him to continue to develop this area. And we also work with Peng Liu at Pittsburgh and to do some computational studies to understand the mechanism better. And we also want to thank the NIH, NSF, and also Stony Brook for their funding support. And finally, I want to thank you very much for all your attentions. And I'm happy to take any questions. Thanks.
And this great talk. Thanks very much, Ming-Yu. And this is a time to thank Ming-Yu for his talk. If you're connected to Mentimeter, maybe Rio can help to give instructions to people on how to join, so that we can get a full range of hearts and thumbs up for Ming-Yu. Okay, so we got a couple of questions, Ming-Yu. So the first is, can you do the cleavage of the bromine carbohydrate with photoredox catalysis?
Yes, we can do that. And so it's not very efficient right now. I mean, we tried to use the iridium and also ruthenium. And so with the ruthenium, it doesn't really work that well because like the excited ruthenium is not reducing enough to reduce the CBr bond. But with the iridium and the presence of the reductants like amine, trialkyl amines, then we will be able to cleave the bromine bond and form the radical. But for that process, it's not efficient at the moment, and it also a little bit difficult to make it broad scope at the moment because we do need the trialkyl amines. And so the trialkyl amine can also serve as a hydrogen atom donor. And so very often what we observe is that we see the reduction of the C2 product, which is one of the projects that I present today. And then, I mean, if you want to go beyond that, then you have to remove the trialkyl amine and using other type of reductants. And we are exploring that and trying to use the metallo photocatalysis and trying to do different type of cross coupling at the C2 position.
Okay, great. And another question, what determines the trans or cis geometric outcome in your nickel catalysis reactions? I think maybe in the arylation reaction. And I was curious also whether you have ligand control potential in that reaction as well.
Yeah, that's a great question. So at the moment, what we see is that the steric play a pretty important role in control the cis and trans geometry, the selectivity. And so at the moment, what we see is that when the C1 acetate, that after it migrate, if we make that group bigger, then we will see more of the trans selectivity. So basically, you will get more of the acyl arylated product. And so that is really the steric at the moment control that. And back to Hugh's questions about catalyst control, we do observe some catalytic control by changing the ligands on the nickel. And so we are still trying to explore and trying to identify the right ligands combinations that will allow us to achieve a high selectivity and also high reactivity. Yeah, thanks for the question.
Okay, great. And then one final one, but please add your questions and then Ming-Yu can answer them in the chat. But one and the questions answered. But one final one from John Montgomery, great talk. Hegui Gong has developed nickel catalyzed reductive cross couplings on C2 acetoxy anomeric bromides with aryl bromides, and that process does not proceed with acetate migration. Why is that? Have you tried reductive cross-coupling conditions with aryl bromides?
Yes, we definitely looking into trying to do the cross electrophile couplings under for our system as well. And at the moment, we don't have any luck yet. It haven't been working at the in this case. And so back to why in his case, like he didn't see that acetoxy migration. So in order for the acetylene migration to take place, there's a few key things that we needed. One is that you need to heat the reaction up so that you can facilitate that migration process. And sometimes, like if you don't heat it up enough, then it may not be able to migrate fast enough. Then what you do is that you get the C1 unrelated product. Another thing is that so the C1, the migration process is the intramolecular reactions, and then the cross-coupling reactions is the like intermolecular reactions. And so in order to bias that, what we did also is that we try to dilute the reaction mixture so that it will be able to favor the migration before it can couple. So.
Okay, great. Well, Ming-Yu, thank you once again for a wonderful talk. There's still lots of questions coming in on the Q&A, and hopefully you can answer them during the next talk.
Sure.
Okay, so please join me in thank Ming-Yu once again.
Thank you.
Okay, so our next speaker is Professor Laurel Schaefer. And she did her undergraduate degree at University of Guelph, and then did a PhD at University of Victoria, and then postdoced at University of Berkeley, and then began a career then back at the University of British Columbia. And she's now she's a full professor then, currently holds a Canada Research Chair in catalyst development. So the Schaefer Group has developed new families of NO-chelated early transition metal complexes for atom economic catalysis to prepare amines, heterocycles, enines, and biodegradable polymers. In particular, the group specializes in organometallic catalysis for organic synthesis, including reaction kinetics and mechanistic investigations. So we're delighted to hear about your work, Laurel. And it's a very interesting title, Head West for Green Hydroamino Alkylation. So welcome.
Great, thanks so much. And thanks for the introduction and the opportunity. I'm really happy to have this chance to share a little bit about how the West Coast of Canada and the west side of the periodic table can contribute to challenges in hydroamino alkylation.
Hi, Laurel.
Yes.
So your display right now is in the presenters.
Oh, really? Okay,
mode.
thank you. How's that? Are we good now?
We are good now.
Excellent, thank you. Now I've got a weird thing going on with this. I'll fix it this way. Okay, so thanks very much for letting me know. That would have been pretty awkward. All right, yes, let me get started here. Many of you are well aware of the importance of amines in a broad range of industries, including the pharmaceutical, agrochemical, and materials industries. And the way that these industries would love to be able to make these amines is by hydroamination, specifically the addition of an amine to a simple alkene. These are very abundant feedstock chemicals, and it would be attractive to be able to do this in one single step rather than the traditional route, which requires first a hydroformylation followed by reductive amination. So today I want to show you an alternative strategy for reacting amines with alkenes. And the reason why I'm here today is because it involves a CH activation event resulting in CH functionalization. So in hydroamino alkylation, rather than adding an NH bond across a CC bond, we actually add the alpha CH bond across the CC double bond. And we can have two regioisomers, as you can see here. This is an attractive alternative to hydroamination. And the obvious question would be, why does this reaction work so well while hydroamination remains a decades-old unsolved challenge? And the reality here is thermodynamics. So a CC single bond gives you about a 10 kcal driving force that's improved over the CN single bond. And you can use that to advantage to overcome the otherwise sometimes thermal neutral hydroamination transformation. Now, my research group interested in early transition metals was very attracted by this early contribution, several decades old now, showing that very simple early transition metal amido starting materials could be used to make this carbon-carbon bond. Temperatures were high, reactions were long, yields were modest, but it was an interesting transformation that actually was pursued by industry for some time before being abandoned. Some other beautiful early transition metal chemistry that resulted were stoichiometric transformations using zirconium, for example, and some beautiful work by Buchwald, Jack Norton, Whitby, and others, who showed that you could actually stoichiometrically make these metalloaziridines do the alkene insertion to make the new carbon-carbon bond. But unfortunately, in these cases, catalytic turnover could not be realized. Around the time that my research group started observing this reaction unexpectedly in our attempts to do hydroamination, Harrison and Hartwig disclosed this communication in JACS that showed that tantalum was in fact a preferred metal for mediating this reaction at higher, with much higher yield. And then the other advantage here was the insight that taking advantage of the arylamine substrate for enhanced reactivity could really improve our approaches of hydroaminoalkylation. So with the inspiration that early transition metals were useful, my research group was interested in pursuing this opportunity. And I want to highlight that this reaction is not unique to early transition metals. The mechanism of early transition metal hydroaminoalkylation is unique to those metals, but there are beautiful examples of late transition metal hydroaminoalkylation that are complementary to the work with early transition metals. In this example here, using a ruthenium carbonyl catalyst, you can actually take advantage of the directing group and oxidative addition to generate hydride intermediates that can then undergo train walking mechanisms to give selectively the linear product. And I didn't highlight this in the previous slide, but Harrison and Hartwig showed conclusively that the branched product is favored. And this has a mechanistic rationale that I'll describe in a moment. In addition to these hydride mediated mechanisms, there's photoredox catalysis that can be pursued as well. So I think hydroaminoalkylation with this range of mechanistic manifolds has the potential to transform the way industry makes amines. And I hope to convince you today that there's some real exciting opportunities there. So as mentioned, we're focused on this west side of the periodic table. We use early transition metals, and we like them largely because not that many of you do. And we're really keen to find out what they can do and what they can do that's different. They're also inexpensive and of low toxicity, so that's very attractive as well. Now, the other question would be why this NO chelating framework? And it has a lot to do with the variable binding modes. If you're working in catalysis, of course, you want to reveal coordination sites for binding substrates, and these four-membered metallocycles are ideal for realizing these transformations. So based on the results shown by Hartwig, we quickly made our first example of a tantalum amidate catalyst with this NO chelating ligand. And immediately we could see that the amidate ligand offered some real opportunities for being able to expand the substrate scope from what had previously been reported, and some interesting observations right out of the gate. For instance, this beautiful diastereoselectivity in setting two adjacent stereocenters in one catalytic atom economic step. Internal alkenes could be used. We got beautiful diastereoselectivity with a bulky substrate like norbornene. So these were the preliminary results that really encouraged us to work towards catalyst development. Now, we are very excited about what hydroaminoalkylation can realize regardless of the mechanistic manifold that you use. But what I would like to highlight is with early transition metals, you have absolutely no additives, you have no co-catalysts or photocatalysts. The reaction is 100% atom economic. The metals are of low toxicity. And there are opportunities for building regioselectivity and stereoselectivity using these catalysts. And that can give us a wide range of potential products depending on the amine starting materials we choose, or the regioselectivity that can be realized in the chemistry. And today I'm going to show you examples of all of these actually. So as I mentioned, the mechanistic manifold in early transition metals is a little bit different. So I want to spend some time here. This is a computational profile that we worked on in collaboration with Eric Lowe. This mechanistic profile has been confirmed using a range of organometallic kinetic experiments. We've also isolated reactive intermediates, kinetic isotope effects, deuterium labeling work. And so this slide is summarizing a lot of experimental effort that I won't have time to talk about today. But what I want to highlight to you is that the electropositive character of the early transition metal is what is really key. So we start out with this pre-catalyst, and what we know from these computational efforts is that the axial and the axial ligands are actually not involved in the chemistry. So I'm just going to remove them from the images here on, but they are still there. So this is a 6-coordinate tantalum center. And the first thing that happens is we have a pre-catalyst that then actually undergoes a protonation event with the amine, eliminating, so this is the amine substrate typically, and this eliminates these L ligands, which can vary from other amido ligands or alkyl ligands, as I'll show later. So now that we have the substrates installed in the equatorial plane, we can actually have the CH activation event that is promoted by the very electrophilic metal center that actually promotes through agostic interaction the reduction in pKa such that that amido ligand can deprotonate and make the metalloaziridine. These metalloaziridines can then undergo the alkene insertion, just like we saw in the stoichiometric chemistry with zirconium by Buchwald many years ago. This 5-membered metallocycle then under catalytic conditions requires the turnover limiting protonolysis step, where the amine substrate comes in and protonates the reactive tantalum carbon bond to make a new bisamido and regenerate the metalloaziridine. And in this computational manifold, we identified that that kappa-1 feature of the amidate ligand was really important for promoting reactivity. So with this mechanistic understanding in hand, we wanted to improve from that first slide that I showed you with the simple amidate. And the first approach that we thought was, okay, let's get rid of the amidos as reactive ligands and replace them with organometallics, because that would just eliminate methane when we activate the catalyst. And that allowed us to realize lower reaction temperatures. We could do internal alkenes for the first time, but unfortunately it was not stable, and you have to make this reagent fresh every time, which is very challenging and laborious synthesis. So we wondered if we could actually control this better by using a ligand on the tantalum precursor. And so by using a phosphoramidate ligand, an alternative NO ligand that is very electron withdrawing, thereby generating a very electrophilic tantalum metal center, we did indeed see improved reactions, but unfortunately this catalyst also could not be stored and was thermally sensitive. But this was our first hint that really by modifying the catalyst, you can get to much more reactive catalyst systems. So from there, we will move to a simple pyridinate ligand. And this ligand that has a very slim steric profile, if you will, allowed for the first example of a broad array of internal alkenes that could be used in reasonable reaction times. From there, my student saw that we could take advantage of commercially available pyridones as a pro-ligand and combine that with commercially available tantalum starting materials using simple syringe techniques to realize hydroaminoalkylation with a range of substrates that have some modest functional group incorporation and allowed for further reactivity. Once again, we saw the beautiful single diastereomeric product when we added an alkylated amine or an ethyl substituted amine to a terminal alkene. This catalyst, however, is limited to terminal alkenes and is not reactive with internal alkenes. So with everything that we had noted so far, we knew that organometallics were particularly useful. So we sought a more robust organometallic, and that led us to this neosylyl derivative that could actually mediate the reaction at reasonable reaction times and temperatures to give quantitative yield. And most importantly to us, this tantalum alkyl can actually be stored as a toluene solvent and delivered, dissolved as a toluene solution rather, and be delivered by syringe. So it makes it much easier to use. With this approach then, we were able to also use some ureate ligands. These NO chelating ligands are more electron withdrawing than amidate ligands, and indeed they do promote enhanced reactivity, where the modular nature of the ureate could be used to advantage to make catalysts with different selectivities. For example, this diphenyl substituted system was useful for terminal alkenes, while the phenyl isopropyl substituted system preferred internal alkene substrates. So these were really promising reactions, and by taking advantage of the electron withdrawing nature of the ureate ligand, we were able to get much shorter reaction times than we had realized so far. So with this ureate ligand, we thought, okay, this is a great handle for installing some stereochemistry by using a chirolamine in the backbone. But as you can see here, the enantioselectivity was far from promising. So that was a bit disappointing, but what was very exciting about this ureate system is that for the first time we saw a broadly reactive system. So I've shown you lots of examples with aryl amine substrates. I've shown you a few examples with alkene substrates. I've shown you very few examples with alkyl amine substrates, but this catalyst can do it. And then the other thing that this catalyst can do quite well is the direct alkylation of heterocycles. So this is a very exciting transformation by, once again, by using simple syringe techniques. We make a stock solution of this sodium salt. We make a stock solution of the tantalum organometallic. We add them together by syringe. And then to that mixture, we add the amine and then we add the alkene. And we can get the direct and diastereoselective alkylation of heterocycles in one simple atom economic step. Now, I promised that I would rationalize why we see this exquisite diastereoselectivity, and this has to do with the reaction mechanism. So if you take the pre-catalyst that I talked about before, and in this case, you're actually activating it with a heterocycle, we make this fused metallocycle that then undergoes the alkene insertion to make the 5-membered metallocycle with beautiful selectivity for the substituent of the alkene to be on the opposite face of the ring. So this is the rationalization for the chemistry that we see. This same catalyst can also be used in combination with more complex and stereochemically refined terpene derivatives. We saw in this chemistry no racemization of this allyl center when using limonene as a substrate. And when using beta-pinene as a substrate, we saw exquisite diastereoselectivity once again to realize one enantiomeric product. This same catalyst can also be used in combination with late transition metals. So early transition metals, as I showed you in the mechanism, do not undergo redox chemistry. And so the aryl halides are compatible with early transition metals, such that we can do hydroaminoalkylation with this arylated styrene derivative. Now, if we were to couple that then with a Buchwald-Hartwig coupling transformation with this nickel catalyst, we can actually then ring close to make indolene products. And notably, If we do it in one pot without isolating the intermediate, we actually get a higher isolated yield after the 2 sequential steps than we do trying to isolate the secondary amine product resulting from hydroamino alkylation. This can then be coupled with a catalytic oxidation that had been reported from Merck, and in one pot, 3 sequential catalytic steps can be realized to generate indolene products in excellent overall isolated yields. This same one-pot synthetic strategy can be employed with these silyl-protected alcohol substrates, such that we can make these reactive intermediates that can then react with tosyl fluoride to deprotect and generate a leaving group, and at higher temperature ring close to make these beta methylated heterocycles, all in one pot, excellent isolated yields. And this can be done on multi-gram scale. We can use the same strategy to make fused heterocycles, where we can take a heterocycle like piperazine, as shown here, to make the same intermediate that can then undergo ring closure. Then, by taking advantage of the fact that ureates can be made into cyclic systems that have the steric bulk tied back, we were able to realize the first example, and the only example to date, of a hydroamino alkylation catalyst that shows preferred reactivity with the less reactive alkylamine substrates. So this could then be exploited by doing a late stage CH alkylation of a Prozac drug to make these kinds of alkylated products in only one atom economic step. No protection of the amine required. We're not limited to tantalum. We can use titanium for very similar chemistry. We've shown that titanium can also be used to make these heterocycles in one pot. In this particular case, we undertook an oxidative deprotection step to make the free pyrrolidine product shown here. We can also take advantage of the increased steric ionic radius of zirconium to use with more sterically demanding amine substrates. And so by using these silylated amine precursors, we were actually able to do hydroamino alkylation, and following workup revealed the primary amine product. So this is the first example of such primary amine intermolecular hydroamino alkylation reactions. And here's an example where we can use the electronic features of the alkene to direct for the linear product selectively. And so Anna was able to show that we could make these alpha arylated amines in one pot using these simple zirconium. It's a commercially available zirconium amido catalyst. More recently, we've shown that you can do hydroamino alkylation of alkynes. In this case, you end up making allylamine products, as shown here. And the real feat in this chemistry was actually realizing catalytic turnover. So we have tried alkyne hydroamino alkylation with all of the catalysts that I showed you before to no effect. But by taking advantage of this tethered ureate ligand on zirconium, we were able to use the 7 coordination environment of this zirconium metal center to actually realize the reactive intermediate required. So using our organometallic approach in my research group, Eric was able to isolate the reactive metalloaziridine. He was able to show the insertion product to generate the 5-membered metallocycle. And you can see here in the 7th coordination site, we actually have the, pardon me, the pyridine unit. Now this pyridine, if it's replaced instead with a protonated amine, could then realize proteinolysis of the zirconium carbon bond to make the desired product. And so this was the first example of zirconium or of hydroamino alkylation of alkynes. With this suite of catalysts now, we wanted to challenge ourselves in material science And so just to wrap up, I want to show you that you can in fact use these same catalysts to realize the amination of polymers. Sabrina has shown that we can do the end functionalization of isopropyl, polypropylene that's vinyl terminated, and using a range of amine substrates, we were able to make these aminated polypropylene materials that display interesting properties. We also were able to couple hydroaminoalkylation with a diene substrate to make an aminated monomer that could then undergo ring opening metathesis polymerization to make the first examples of controlled polyaminated materials. We are able to use the pyridinate catalyst to selectively monoaminate these strained alkene rings, which can then undergo ROMP polymerization. And I want to highlight here, this is a nice example that we can actually do these reactions on very large scale. And so what was really surprising to us is that we were able to make, and this video was working earlier. I don't know why it's not working now. Oh, that's too bad. These are self-healing materials, and this video actually shows these two spheres when they're put in contact, they autonomously heal with no temperature, no pressure, and no solvent. And this is shown quantitatively here. The virgin material is the black. This is a stress strain curve. And if we allow it to heal for only 5 minutes, we can see that it's recovered substantially. And in only 20 minutes, a polymer of about 25,000 Dalton actually fully recovers to its original strength. So these are pretty interesting materials that we are exploring. So with that, I hope I've given you a useful and interesting introductory tour of using early transition metal hydroaminoalkylation for making a broad range of organic products. We can use this in materials synthesis. We can use group 5 and group 4 metals, and the NO chelating framework is key for realizing these transformations. I want to thank the people who've really done the work. I work with an amazing team of co-workers who have been remarkably resourceful during this very challenging time. I want to thank these organizations for funding and contributions, you for your attention, and I'd be happy to answer any questions.
Very nice indeed. And I'm going to start the let's see, I'm going to start the positive responses myself to just to lead the effort. Very excellent material. That's lovely. Okay, so we've got a question from Shengying Xie saying, thanks for a great talk. I'm wondering the bis or multifunctionalization didn't occur, or much less in heterocycle substrates. Could you explain it? Thank you.
Yes, because of the mechanism of our reaction, steric inhibition by substrates is really quite profound. And this steric inhibition results in this beautiful selectivity for monoalkylation. In fact, an area of catalyst development in the group right now is to realize catalysts that are able to realize multiple alkylation events. But all of our data is consistent with just steric control.
Okay, so tell me why those. This is a non-central part to your talk, but why do those polymers recover like that?
Yeah, this is a really fascinating property. So the polyaminated materials are what are belong to a family of materials called associating polymers, which have dynamic non-covalent interactions. And so you can imagine just like two drops of water that touch, they immediately become one drop of water on a much slower time scale. The amines of the polymer with the same hydrogen bonding interactions actually induce the same kind of behavior. And so we've shown this by, first of all, preparing tertiary amine containing polymers. They do not self-heal. So you remove the NH functionality and that feature is gone. The other thing that we've been able to do is show that by incorporating more than just the amine hydrogen bond acceptors into the compound, but other ether functionalities, for example, fluorine functionalities even, we're able to modulate the associating behaviors of those materials and the self-healing behaviors of those materials. So we attribute the self-healing behavior to the hydrogen bonding character.
Okay, great. All right. So, well, once again, thank you for a lovely talk. And if there's any more questions, please add them to the Q&A, and Laura will continue to answer them offline as well.
Great.
Okay. Well, thanks very much again, Laura. Very
Thank
nice
you.
indeed. Okay, so we're moving to our final speaker. This is Ming-Yu Ng from the University of Toledo. And I think we're going to have another angle on how you can functionalize, do CH functionalizations involving amino compounds as well. So very different approach coming up. So Mike did his undergrad degree at Western Carolina, then did his PhD at Southern California. And then began his serious studies related to CH functionalization because he went to work with Professor Guangbing Dong when he was at Texas at Austin. And since then, he's moved to the University of Toledo. And I actually had the great fortune of hearing Mike for the first time at a Pacificam meeting just recently, a virtual talk. And I was really blown away with the program that he's developed. It's a very exciting program. And so I'm really excited to hear more about it, Michael. So go ahead.
Thanks, Hugh. And just give me 1 second. I'm actually having trouble. Everything was working well before we started, and now nothing is working.
Okay, that's fine. You're doing better than me. Usually everything's working badly all the time with me. So you're slightly ahead. So we can wait for you. Don't worry.
Problem is I can't actually see the screen that I want to share to.
Rio, have we got the slides? Can we share the slides?
No, I don't think I had sent them over.
If you want to send them over, we can do that as a backup as you're trying to get sorted out. Unless they're like my slide deck, which is probably 250 megabytes worth of stuff.
Okay, I think.
That looks good.
Do you see?
Do
you see nothing on the screen?
No, we see the screen. You just got to put it in projector mode.
I was just making sure that you see anything on the screen.
Yes, we do.
Okay, so that probably is not the right one then. Now you see it.
Yes, that's perfect.
All right. Sorry about that.
It's always good to have a little bit of excitement. I think it always helps to get the blood pressure going, you know. Okay, all yours.
Well, the good news is there's not 1,500 people here, so this isn't quite as embarrassing as if there were. All right. Thank you again, Hugh, and thanks everybody for sticking around for the last talk. And so, as Hugh said, we're really focused, or spend a lot of time talking about functionalization of the amines, maybe a slightly different approach to what you've just seen. Now, this is the obligatory, this is the work that's being done in my group, and mostly what I'm going to talk about today, or entirely what I'm going to talk about today is some of our work on trying to come up with new strategies for functionalizing amines without having to use any directing groups for late transition metal functionalized catalysis. There are also a couple of other things such that as trying to do CH carboxylation that I won't talk about, although you'd sort of imagine that this would be pertinent to today's talk. And so I think for this audience, I don't really need to explain this, but amines are prevalent throughout drugs. And so this is sort of why we're interested in this chemistry. And quite specifically, beta, gamma, and delta arylamines are especially prevalent. And here I've just shown several examples of gamma arylamines and biologically active compounds. Now, when I started my position in 2016 at the University of Toledo, what was really one of the things that was really ramping up was the opioid epidemic. I don't know particularly when that term came out, but it's really when it started to hit sort of its crescendo. And so you can see that Ohio and West Virginia we're #1 and #2 in the country at something. Unfortunately, it's not quite what we would like to be winning at. And so from the perspective of a chemist, is there anything that we could do about this? And what we think is that a lot of opioids are very potent, and that's one of the reasons why they're popular analgesics, but the challenge is that they also have relatively narrow therapeutic indices. You can see a lot of examples here of opioids that you probably know by name, and you can see the therapeutic windows are relatively small. So what we thought was something that we could contribute to this is coming up with new ways to maybe synthesize either unnatural morpholanes or even apply this type of chemistry to other potential opioid receptor agonists. So that's really where we got our start. Now, you've seen obviously lots of ways to do amine functionalization, but especially when we're talking about late transition metals, I just want to back up a bit and say that historically, free amines are not usually very good directing groups for late transition metals. And a key reason for that is that they'll commonly undergo beta hydride elimination. And so Gaunt especially has dealt with this problem by making sure that you have very sterically bulky amines that are unable to undergo beta hydride elimination. And so this works very well. You don't need any additional directing groups. You can use the amine directly. The challenge is now that you've got a very sterically bulky amine, and overall the substrate scope may be a little bit limited. Now, if you expand your scope to other types of CH bonds, for example, benzylic, or aromatic CH bonds, this becomes a little bit easier. But again, for aliphatic amines, this is still a bit of a challenge. However, sort of the more traditional approach is to instead convert your amine into some type of an amide. And the benefit of doing this is now that you've made the amine much more chemically robust, so you don't have to worry about oxidation procedures or background reactions. But the challenge is now you've got to install this directing group and later remove it. One thing that's important to consider is if you're making a new carbon-carbon bond, this is usually a trivial thing to do, but if you're making something more labile, you have to worry about whether or not you can deprotect the amide as well as maintain your functional group. So about the time that my group got started, the idea of using amines as directing groups for amine functionalization really took off. Now, this idea is certainly not new, but the idea of using these transient directing groups as catalytic directing groups, despite having been known for carbonyl functionalization for 30 years, had really only just started being applied to functionalization of amines. And this is a wonderful approach, but it does still pose a couple of challenges. First of all, you still have, especially if you're using your directing group catalytically, free amine in the reaction, and that's going to be sensitive to oxidation, and it can also serve as a nucleophile. Meanwhile, you've got a metal coordinated imine in the reaction, which is sensitive to different modes of oxidation, as well as now it's a somewhat active electrophile. And so while this works under maybe mild conditions, we were concerned about whether or not this would actually be useful for reactions that would require harsher conditions. So when we got started, we wondered if there might be a way to get the benefit of both of these. Could we do something that's transient like an imine, but also more chemically robust like an amide? I think really I'd be remiss not to mention inspiration that we got from LaRosa, Leitner, and Yu on this, where we thought, could we actually use something like a carbamate? And if we could use a carbamate, could we actually use a carbamic acid directly? And in this case, we could just add CO2 to our reaction, form the carbamate in situ, run the reaction, and then at the end of the reaction, bubble off the CO2 to get to our free amine products. And it turns out this actually works relatively well. And so here's our first foray into this. And so we could take tert-amylamine as a model substrate, different aryl iodides, palladium as a catalyst. We need a silver additive to activate the CI bond and potentially sequester the iodide afterwards. And this works on a variety of different electron-rich and electron-poor arenes. Now, this was work that was done by my first postdoc, Dr. Kapoor, and you'll notice Daniel. So Daniel was 11 years old when he joined my lab and started doing this work. And I just like to highlight that if an 11-year-old can do this, I think it's reasonably robust chemistry, even if he is, much smarter than me, 11-year-old. Now, there's an elephant in the room here. You'll notice that I've said 1 to 2 equivalents of CO2. And you might wonder, how does one measure 1 to 2 equivalents of CO2? And we had considered trying to do this with a syringe or with a balloon. But what we realized was the easiest approach is to simply add dry ice to our reaction and then seal them. The benefit of doing this is we can actually build up mild pressures. And I'll let my students explain that in a little bit more detail.
The key to success is to ensure that the appropriate amount of dry ice is added in a timely manner and that only dry ice, not deposited water, is added to the reaction vessel.
After weighing out 26.3 milligrams of dry ice, immediately add the ice and seal the vial with a PTFE-lined cap. Then transfer the reaction vessel to a preheated 110 degrees Celsius plate for a 14-hour stirring incubation. The next morning, open the vial after cooling to vent the CO2.
I have to say my favorite part about this chemistry is that I have a soda or pop habit, depending where on the country you find yourself. And so I really always enjoy opening these. It reminds me of opening a brand new Coke. That's for the folks at Emory. And so it turns out that we can also do this on other types of amines. So alpha tertiary amines, of course, should all work, and they do. But what would be more challenging is something where we now have the ability to do oxidation of an alpha carbon. And it turns out just by lowering the temperature, we can now also do alpha secondary amines. Unfortunately, alpha primary amines still don't work in this chemistry, we think, simply because of conformational bias. Now, one of the things I didn't mention is that one of the issues with transient directing groups is typically they don't work for secondary amines. They're only going to work for primary amines. And so what we thought CO2 might allow us to do is now enable this chemistry to work for secondary amines. And it turns out it does work. The yields are a little bit more modest, and we have to dramatically increase the loading of CO2 to achieve this, but we are eventually able to get these reactions to work. Now, this works for acyclic amines, and we thought, oh, it'd be brilliant if we could expand this to cyclic secondary amines. And initially this looked very promising, and we thought we had about 12 substrates, and we were really excited about this until we really dug into the NMR data and realized that we seem to be missing this CH at the alpha position. And after not very much time, we realized that what we're actually getting are these partially oxidized products. Now we're certain that the CH aerylation occurs first, but even under these conditions, we're simply not completely able to shut down oxidation of that CN bond. And so in this case, we're able to get a couple of substrates. Again, some of them, the yields are not particularly good. So we're still trying to figure out if there's a good way to improve this chemistry. I will say there is a silver lining to this, which is that we can take this racemic starting material, get to these partially oxidized products where now we could do asymmetric hydrogenation to actually get into some potentially useful natural products. So in terms of the mechanism, I just want to point out that CO2 is probably not stoichiometric, or isn't required to be used stoichiometrically. So we can make this internal salt, and then we can see better than stoichiometric conversion based upon it. We also find that if we run the reaction under deuterated media, we don't see any deuterium incorporation, but we do see if we run the kinetic isotope effect experiment that we do have a positive KIE. So CH activation is rate determining. Now, the one thing I haven't really discussed and won't really get to say too much about is exactly what is the role of CO2 in this chemistry. And so one thing that was proposed to us is that perhaps it's just allowing us to take one of these palladium dimers and disperse it. Well, because we're in acetic acid, it turns out that's not necessary. This can actually disproportionate on its own. So we don't think that's necessary from the perspective of the catalysis. Unfortunately, if we simply take the dimer and try to treat it with CO2, in many solvents, CO2, CdCl3 being a representative example, we don't really see any formation of carbamates. Typically, though they are known, they're formed at much lower temperatures, and we're obviously at a much higher temperature. One exception is that if we do this in DMSO with some acetic acid, we do begin to see new resonances in addition to the dissolved carbon dioxide. So we can also see some new things. Unfortunately, we can't isolate anything. So we decided to turn to mass spec. Now, before I get to the mass spec, I'm just going to put up a catalytic cycle that I don't mean. But we had spent a lot of time trying to think about what this might look like. Could it be a 7-membered metallocycle? And at the time, some work came out from Dagong Yu's group that actually showed quite nicely that actually a 9-membered metallocycle with 2 equivalents of CO2 for a slightly different system was actually about the same energy as a 5-membered metallocycle with no CO2, whereas the 7-membered metallocycle is actually quite higher in energy. And so when we started doing our mass spec studies, we found several palladium species, but one of them was this. And if you squint really hard, this looks a lot like this 9-membered metallocycle with loss of acetate. Unfortunately, after further exploring this, we realized it's actually just this bisamine complex where one side has undergone CH activation and the other is not. So we're still trying to figure out exactly how to go back and probe the mechanism of this. So one thing I just want to point out is my postdoc thought, well, we should try to extend this chemistry to benzylamines. And in my head, I thought this was a solved challenge. And so there's not really a reason to do this. And it turns out if you're looking at tertiary benzylamines, then this is true. There were several catalytic procedures, but actually for primary and secondary benzylamines, there were very few. And a key is that generally speaking, you didn't get free reactions out because the reaction conditions are fairly harsh. Now, in this case, obviously, you're intentionally doing the lactamization, but in this case, you're simply reacting with a solvent because the conditions are fairly harsh. And so we wondered whether or not we could just toss in some CO2 to this type of a reaction and make it milder. Short story is, yes, we can. So this works very well for a variety of primary and secondary means, in some cases with some interesting functionality. We could even install this quite complex group. Now, you'll notice that all of these arenes have a functional group at the 2 or 3 position. And this is by design. I think everybody here probably recognizes that if you don't block those positions, you'll get diarylation. And that was certainly true in our case. But we did find some notable exceptions to that. If we have some type of an amine where there's a beta coordinating or chelating functionality, we can now realize these reactions monoselectively without the need for any appended functional groups on the ring to block it. Obligatory, we were able to make the key bond in a nastrepib. Unfortunately, it failed its phase 3 clinical trials, so now nobody cares. All right, in terms of the mechanism, we tried to see if we could prepare the deuterated substrate by reversible CH activation, and that doesn't work. So we had to come about it a different way. And then we could do the KIE study to show that, in fact, CH activation is reversible. And so we decided if CH activation was reversible, that if we vary the concentration of CO2, we should see first order rate dependence for it as well. And it's very interesting that we do actually see first order rate dependence at low loadings of CO2, but then there's actually a second inhibitory effect. So again, this suggests that CO2 is involved in the CH activation, but what else could be going on here? One of the things that we're very interested in is actually doing CH carboxylation. So we wondered if that might possibly be going on in the background. And so what we did was we prepared the product of that, ran the reaction. And what we see, interestingly, is not only do we not get any aerolation product, we actually get full decomposition of the starting material. And so we think basically as we increase the loading of CO2, basically this forms, and then in the presence of silver, it will then decarboxylate and then ultimately shuts down our palladium catalyst. And so that's why as we increase the loading of CO2, we more quickly kill our catalyst. At least that's our working hypothesis. There is a slight induction period. Not an induction period. There's a slight period of faster reactivity, and then it slows down. So again, that sort of tracks with that as well. All right, so in terms of the mechanism, it's pretty well understood that palladium gets to these types of dimers with benzylamines, and what's less understood is how those turn over into product. And so our hypothesis is now that something like this is happening. Again, we're still working on trying to tease out exactly what some of the intermediates are. But just to confirm that, we do make the dimer and then run our key reaction with it, and we see at least under for this substrate that we don't actually get any product. All right, so we also tried this with ortho coordinating functionalities, and we do get product, but they don't stop here. They actually cyclize. But the other thing that's quite interesting is it turns out we don't actually need CO2 anymore to make these work. And so what we think is that if the challenging step is the oxidative addition, the ortho-chelating functionality simply lowers the barrier for that. So we don't really need as harsh conditions anymore. Based on that, we wondered if we could use harsher oxidants, if we could actually also get turnover without the need for any type of added directing group. And it turns out this works very well for chlorination. Bromination works, but the competing electrophilic aromatic substitution's pretty good if you've got an electron-rich arene. And then we can, if we try to do fluorogenation, instead, we actually get reductive elimination of the solvent rather than the fluoride. All right, so very quickly, I want to talk about how we've tried to expand this chemistry to allylamines. And at first, we wondered if we could do just simple CH activation of these substrates, recognizing that there's several other side reactions that could possibly occur. But unfortunately, because this is could potentially form a pi allyl, there's also several undesirable reaction pathways as well, where we don't even install an arene. And it turns out that we actually see all of these depending on our reaction conditions. However, I have a very brave master student who was able to push this over the goal line. And so we were ultimately able to show a reasonably nice substrate scope for this. Generally speaking, the Z to E selectivity is pretty good and obligatory drug synthesis. Now, it turns out that all of these are oils. except for this this product. And eventually we decided, well, let's try to crystallize it. Unfortunately, when we did that, we realized that we've misassigned all of these. And so rather than these being products of CH activation, they're actually products of an insertion reaction. And that was a little bit surprising to us because typically you don't tend to make tri-substitute alkenes via insertion reactions. So we're pretty excited about that. If we could do this from the synamylamines, could we do it from the terminal alkenes simply by increasing the amount of coupling partner? It turns out the answer is yes. Although if we then try to go bigger and do the reaction 4 times, the efficiency does drop quite a bit. We can also scale the reaction reasonably well. Now this is an unoptimized 54%. All right, so since this is an insertion mechanism, we wondered, could there be any chain walking? And the answer is there isn't actually any chain walking. But then that really led us to the question of, okay, well, if this is an insertion reaction, where's that other isomer coming from? And quite interestingly, despite significant other literature suggesting that this is possible, for these specific substrates, we don't actually see any isomerization. So if we take, if we can isolate one of these products and resubject it to our conditions, there's no isomerization. So that really suggested to us that these 2 products are coming from different competing pathways. Maybe one is an insertion, but maybe we're also seeing that CH activation that we expected. And so I won't talk about why we can't do it right now, but it turns out kinetics for these reactions are not very reproducible. So what we wondered was, could we instead look at the product distribution sort of as a proxy for the kinetics? And so if we take the protease substrate, run the reaction under conditions that give us a 1-to-1 mixture of those 2 isomers, what would happen if we then ran it with the deuterated substrate? And what we see is that if we use the deuterated substrate, the yield drops, but it's entirely because we are slowing down the formation of that cis product. And so that really to us suggested that these 2 isomers are actually coming through different competing pathways. The other thing I want to mention is that although we had been using CO2 in all of these cases, it turns out it's not really necessary. It gives us a better yield if we have it, but it's not absolutely required. And again, the kinetics here we can ignore. What is consistent is that in the absence of CO2, the overall decomposition of mean in the system is much faster than if we have some CO2 present. And so that really suggests that all CO2 is doing is just helping to protect off-cycle amine from decomposition. The challenge now is that with CO2, our regioselectivity made perfect sense. But in the absence of CO2, that would now imply that we're making a 4-membered metallocycle, which via an insertion reaction seems unlikely. So coupled with the fact that our kinetics were not very reproducible, we started wondering, could it actually be some type of palladium nanomaterial that's actually responsible for this? And so we decided to run our reaction with some mercury, and that seems to shut down the reactivity. So that might suggest nanoparticles. Another thing that we had done was we actually just preformed some nanoparticles and then ran the reaction. And it worked reasonably well. So on the basis of these experiments and some others I don't have time to talk about, we really think that there's 2 competing processes here. One, which is an insertion mechanism catalyzed by nanoparticles, but there's also this background CH activation reaction. All right, I'm going to skip this because I think I got started slowly. And so the last thing I want to talk about just to finish off is to talk about where are we going with this work. And so what we'd really like to be able to do is to have complete control over where we can install functional groups based maybe on the catalyst speciation. Now, we're pretty confident at this point based on several experiments and projects that we've started that a lot of these insertion mechanisms are actually going through palladium nanoparticles. But the CH activation then is challenging if our substrates are readily decomposing a little bit to give us those nanoparticles. And so we're really inspired by some work from Gaunt here, where we wondered if we could flip from, say, palladium acetate to something that's ligated and maybe has more preference for CH activation versus beta hydride elimination, could we now render these CH activation reactions competitively? And so this was sort of the idea there. Maybe we need CO2, maybe we don't. And just to give you an idea that this actually does work. And so if we take this synamylamine substrate, in this case, we add various monoprotected amino acid ligands and now switch our coupling partner to boronic acid, we can actually get this at up to 70% yield. We're still working on exactly why tempo is important for this, but it does increase the yield by about 30%. With that, I would like to thank you for your attention. This is a slightly old picture of my group. And I would like to happily take any of your questions.
Okay, excellent. So a very nice talk. You've been very creative in the different ways you can exploit this chemistry. It's really pretty impressive. Okay, so we have a question from Kirk Shimkin, who says, for cyclic amine substrates like cyclohexylamine, can you functionalize the carbons on the ring?
So if we have a simple cyclohexylamine, the answer is no, at least not very well. What we have found is we have, I guess I didn't dote on it when I got there, but so we're able to take, for example, norbornylamine type substrates, and then we can actually do methylene CH activation on those. But we typically need a fairly rigid ring system to be able to achieve that.
Okay. So another question for you, and this is from Laurel. You can ask the question yourself as a speaker, so you have the privilege.
Great, thanks. Thanks very much for a really nice talk. Very much enjoyed it. In the latter work where you showed this rather unexpected observation that tempo is really important, that suggests to me that nickel may actually be particularly useful for some single electron chemistry. Have you given it a shot yet?
We have, I can't say that we've given it a good shot, but I'm always, I'll tell you what, palladium has tripled in price since I started my position. And so I'm looking at your side of the periodic table these days, as much as possible. We've tried several things with nickel. Unfortunately, at least for the things we've done, it never quite works as well or at all in some cases.
Okay, thanks.
But it's definitely on our radar, just not successfully yet.
We got a question from Shenghing She. Thanks for the talk. In your previous conditions with more CO2, decarboxylation was found, but it differed in the latter reaction with alkenes. Could you explain more?
Sure. So the different reactions, so CO2 has potentially different roles across this chemistry. So for the alkenes, CO2 is not really involved as a directing group. We don't think there's any role for it. We did for a long time due to some very dodgy control reactions that didn't work very well. We still don't really know why those didn't work. And then suddenly the control reactions did work. But we're fairly confident that CO2 serves no role as a directing group. It's just serving as a protecting group. Whereas for the benzylamine chemistry, we think that in that case, you're actually able to, one, CO2 is actually serving as a directing group there, but then also it is able to reductively eliminate.
Okay.
So that answers the question.
Good. And one final question. Have you made carbamates with nitrogen as a part of the ring, like pyrrole?
We have tried doing that, and especially to see if we could do some functionalization using that approach, both on pyrrole and indoles. Unfortunately, at the time that we were doing that, it was all under acidic conditions, and what we found is, at least it seemed like to us, that those readily decarboxylated, and so then they just polymerized. So we didn't really get any products out of that. Now, what we would like to do at some point is go back under more basic conditions and see if we could promote that chemistry, but we haven't gone back and tried recently.
Okay, great. Well, thanks very much. Great talk. And now I'd like the audience to join me in a really enthusiastic Menti thanks to the speakers. If you're still connected up to Menti, this is your time to show us you're connected. Okay, great. Well, congratulations, everyone. It's been a wonderful collection of talks and really showing a lot of really interesting new directions for CH functionalization to continue to develop. So once again, thanks to all the speakers, and hopefully you'll join us when we have our next future meeting as we continue the tradition of these virtual symposia. Thanks very much, everyone. Bye now.
Thank you.
Right.